This is the first of a multi-part lecture series that walks you through the developmental processes that an embryo undergoes from fertilization to organogenesis. During this 8-week time period, the embryo transitions from a single cell into a fetus that is recognizable as a baby. The current lecture will concentrate on the events that take place during the first week, namely fertilization and implantation. Fertilization occurs when a sperm from a male unites with an oocyte or ovum from a female. At this point, a zygote or fertilized egg is formed. In order for the sperm to gain access to the ovum, it needs to fight its way through a few layers before making contact. The most notable of these layers being the Zona Pellucida. The Zona Pellucida is a membrane that surrounds the ovum and contains sperm receptors which attract the sperm. Once the sperm fuses to the ovum, the Zona Pellucida hardens to form somewhat of a shell around the ovum. The shell is advantageous in that 1, it helps to prevent implantation in the uterine tube, and 2, once fertilization occurs, the sperm receptors on the Zona Pellucida begin to break down so that additional sperm are no longer attracted to the already fertilized ovum. This avoids polyspermy, or the fertilization of one ovum by multiple sperm. The illustration within the box depicts a zygote after a single mitotic division. Notice the ring that encloses the two cells. This is the Zona Pellucida. Fertilization is followed by a rapid series of mitotic divisions. Because the cells are still encased within the now hardened Zona Pellucida, there is no period of cell growth between divisions. Instead, the large parent cell continually divides into two smaller cells, and then from there to here. This unusual pattern of cell proliferation is termed cleavage, and the cells produced are referred to as the blastomeres. Once the embryo undergoes 4 rounds of mitosis and consists of about 16 cells, it is now referred to as a morula, and this is due to its resemblance to a mulberry, which is what the name means. With a few more rounds of mitotic division, the morula gives rise to a blastocyst. This process occurs as the cells of the morula begin to differentiate into 2 distinct masses. Cells surrounding the perimeter of the morula differentiate into an outer cell mass, while those forming the core of the morula begin to sequester themselves at one end and become known as the inner cell mass. The outer cell mass is later termed the trophoblast, and these cells will contribute to the formation of the placenta. The inner cell mass, otherwise termed as the embryoblast, give rise to tissues of the embryo proper. Also take note that with the differentiation of these 2 masses, a cavity is also formed, referred to as the blastocele. Prior to formation of the blastocyst, meaning the differentiation into the inner and outer cell masses, all cells, all of these blastomeres, are totipotent. This means that each cell has the potential to develop into all tissues necessary for fetal development. This includes both embryonic and placental tissues. Following blastocyst formation, however, the differentiation into inner and outer cell masses limits the type of tissues these cells can form. Cells of the interior are limited to forming only embryonic tissues. Cells of the exterior are restricted to forming cells of the placenta. Internal cells are said to be pluripotent, meaning that they are capable of giving rise to many types of cells, but not all those required to support fetal development. With additional differentiation events, each cell's potential fate becomes increasingly restricted. Once the developing embryo reaches the uterine cavity, a small hole develops within the zona pellucida, which allows the blastocyst to hatch from its shell. The freed blastocyst is now able to attach itself to the uterine wall. The picture on the left shows this process occurring. Here you can see a portion of the hatching blastocyst as it begins to protrude through a small hole in the zona pellucida. If, during the process of hatching, the blastocyst becomes squeezed in such a way that the embryo splits, the embryoblast splits, then monozygotic twins will result. And monozygotic simply means that two babies were produced from a single zygote. If the embryoblast pinches off but does not undergo complete separation, then conjoined twins will result. Most of the time, however, the embryoblast hatches unscathed and a single baby is formed. An additional possibility associated with twinning is the formation of dizygotic or fraternal twins. Dizygotic twins result from the ovulation and subsequent fertilization of two mature oocytes. This illustration depicts transport of the ovum and early embryo down the uterine tube and into the uterus. Fertilization typically occurs in a distal portion of the uterine tube in an area referred to as the ampulla. Fertilization may occur up to 5 days after intercourse. The transporting zygote undergoes cleavage, becomes a morula, and then develops into a blastocyst, all within about 4 days post-fertilization. Once the blastocyst is within the uterine cavity, it frees itself from the zona pellucida, as evidenced by these faded dashed blue lines, so that it can begin to embed itself into the uterine wall. Implantation occurs around day 6, with the embryonic pole of the blastocyst, or the area of the blastocyst with the inner cell mass, burrowing its way into the endometrium. By the end of week 1, implantation has begun. To facilitate this process, the cells of the trophoblast are beginning to differentiate into 2 layers. The cells lining the blastocyst cavity give rise to the cytotrophoblast, whereas the cells lying external to the cytotrophoblast at the embryonic pole fuse to form the syncytiotrophoblast. The term syncytium simply refers to a mass that contains several nuclei as a result of multiple cells fusing together. And you can see this within the syncytiotrophoblast layer as evidenced by the presence of several nuclei. Functionally, the syncytiotrophoblast is a highly invasive tissue which begins to erode its way into the endometrium. At the same time that the trophoblast cells are differentiating, the embryonic cells also begin to separate into 2 layers. The embryoblast is starting to organize itself into a 2-layered bilaminar disc. The superior portion becomes known as the epiblast, whereas the inferior portion that is closest to the blastocyst cavity or blastocele forms the hypoblast. This marks the end of week 1.